Welcome, everyone, and thank you for joining us. My name is Alana Heckler, and it is my pleasure to welcome you to the fourth episode of the Infectious Disease webinar series, hosted by the Euroimmune Academy. We are excited to present today's topic, measuring T-cell responses and its application to the study of SARS-CoV-2 infection and vaccination. Following the presentation, we will open the webinar up for a Q&A session. If you have any questions for our speaker at any time, please type them in the chat box on your screen, and we will relay them at the end of the talk. If you are interested in claiming PACE contact hours for this webinar, it is important that you log on individually and stay for the entire duration of the webinar. This will be counted as your attendance. After the webinar, you'll receive instructions on how to receive your PACE certificate. Without further ado, it is my pleasure to introduce to you our speaker, Dr. Alessandro Sette. Dr. Sette is a professor at the La Jolla Institute for Immunology and heads the Center for Infectious Disease and Vaccine Research. Dr. Sette graduated as a doctor in biological sciences from the University of Rome and did postdoctoral work at the National Jewish Center for Immunology and Respiratory Medicine in Denver, Colorado. He founded Epimmune in 1997, where he served as Vice President of Research and Chief Scientific Officer until 2002, when he joined the La Jolla Institute. Dr. Sette has devoted more than 35 years in biotech and academia to understanding immune responses and developing disease intervention strategies against cancer, autoimmunity, allergy, and infectious diseases. Dr. Sette's laboratory is the world leader in the study of epitopes, and he has overseen the design and curation efforts of the National Immune Epitope Database, a freely available, widely used bioinformatics resource. His lab uses knowledge of epitopes to define the hallmarks of a beneficial immune response associated with effective vaccines, as opposed to immune responses that are ineffective or that cause harm. Dr. Sette, we are honored to have you here today to discuss the utility of measuring T-cell responses. And with that, I will hand it over to you to begin your presentation.
Thank you, Alana. What we are going to do today is talk about measuring T-cell responses and importance in SARS-CoV-2 infection and vaccination. And I'm going to break down the talk in two parts. One, generally speaking, about T-cell responses and issues about its detection and assays and so forth, and then go into more depth in the relevance of T-cell responses for COVID-19 and SARS-CoV-2 vaccines and so forth. So T-cells, and before we dive into T-cells, I just want to give a little background on the different types of adaptive immunity. So there are three main types of adaptive immunity, a little bit simplifying here, but one are the antibodies. And antibodies are key in human vaccines. They are the ones that prevent infections or reinfections. So neutralizing antibodies, what they do, they bind to a virus and basically neutralize it. But if an infection occurs, the virus at that point hides inside the cells of the host and becomes unavailable for recognition by the antibodies, which are soluble proteins. And so you have this second line of defense, the T cells. So T cells come in two flavors. One are the helper T cells, or CD4 T cells, which, as the name says, are helpful. And what are they helpful for? They really are crucial to orchestrate the development of antibody responses. Without helper T cells, you don't get a good high affinity IgG response. But beyond that, also the helper T cells are key to having antiviral effect. They can have direct antiviral effect. They also can be cytotoxic, and so they have also independent activities. And say CD8, CTL, cytotoxic lymphocytes, they're killer cells. As the name says, they are crucial. They kill infected cells, and so collectively the helper and killer T cells are not very effective in preventing the infection, but they're very crucial to modulate and terminate the infection and play a role in modulating disease severity. So we are particularly interested in measuring T cell responses for the purpose of evaluating vaccines and study immunopathology. Now, one problem is that measuring T cells, it's significantly more challenging than measuring antibody responses. Not that antibody responses are easy to measure, but T cells are generally more difficult because they are live cells and they are a little bit more finicky. And as I will discuss in a second, there are different assay methodologies, each one with its own advantage and disadvantage. And in general, there is a tension here in the sense that the easier the assay, usually the less information you get. And the more sophisticated assays at the other end of the spectrum are much more cumbersome and costly, but deliver a lot more information. So this is just from earlier in the year. There was a letter from several prominent scientists to the FDA, really highlighting the fact that it's important to measure T cells. This was in the case, of course, of COVID vaccines and infection. We'll get back to that later on. But in general, T cells are not as routinely measured, and this is a problem because you get incomplete information if only the antibodies are measured. So what kind of assay methodologies are available to measure T cells? Well, whole blood assays called quantiferon assays or something of that nature are very simple. To just basically add whatever stimulus to whole blood, and then you measure interferon gamma release or other tests. But the problem is least informative because you just measure a response. The assay is simple, but you don't know whether the response is coming from CD4, CD8, what else might be going on. ELISpot assays are robust and relatively high throughput. They have a little bit more work that has to go into. Also, they still do not provide information about the phenotype of respondent cells and are also limited in sensitivity as compared to other assays. And ICS, intracellular cytokine staining, what this assay does is it measures, as the name is saying, by fluorocytometric methodologies, the particular cytokines that are produced by different cells. This is very, very sensitive, and also because it's a cytometric fluorocytometry assay, gives you the information of what the cell does, what other markers it may be expressed, and so forth. The drawback of this is that you're measuring a particular cytokine. So if you're measuring interferon gamma, what if the particular T cell you're interested in doesn't make interferon gamma, but makes IL-5 or makes leukin 17? So the other assay down here, this AIM activation induction marker assay, was specifically developed to overcome these issues, and it's agnostic because it basically measures when T cells are activated by antigen, regardless of what they do or they don't do. Is also, again, is a flow cytometry assay and allows to capture the cells response alive. So you can do downstream analysis, mRNA profiling, and so forth. And then there are other assays such as, for example, tetramers or multimers or TCR sequencing. And in general, tetramers and multimers are gold standard in terms of how detailed in information they yield, but at the same time, they are limited by the fact that you only get information about one particular peptide HLA combination, and we'll get back to that in a second. So as I was saying, we like this AIM assay a lot. This was developed mostly by Shane Crotty's group, and we collaborate to those studies, for the purpose of detecting follicular helper cells, TFH. These are very important cells that are in the germinal centers, and they really are the key cells that are involved in developing an antibody response. And they work by contact with B cells, so we don't secrete much in terms of cytokines. So if you use ELISpots or any cytokine release assay, you're blind to the cells. They're very stingy in terms of producing cytokines. But if you use this assay where you look at the cells that are activated by various different activation markers, they become readily detectable. Another issue related to T-cell assays is whether you're going to do an assay with just fresh cells or previously frozen but still freshly thawed cells directly ex vivo. And this is the most physiologic. There is minimal alteration of the phenotype, as again can be combined with transcriptomics, but in a way is as a disadvantage. You require relatively more cells because you need to see a signal right there, and so it's less sensitive, relatively speaking. The alternative is to take your cells and culture them for 1 week or 2 weeks. This expands the number of antigen-specific cells. And so by definition, this allows detection of very weak responses, is also good for TCR repertoire determination, just like the one up front, but require longer assay time. It's a lot more laborious and expensive. Certain cell types, like regulatory T cells, do not expand well. And the phenotype is altered because the in vitro stimulation obviously changes all the phenotype of the cells because they become activated and proliferate. And in fact, some cell may expand better than others. And a lot of studies that report narrow TCR repertoires or epitope repertoire are employing in vitro stimulation, presumably also because there is a bias in which cells expand. So as I was saying, we covered which assay, different assays are available, which modality, ex vivo or in vitro stimulation, and all this situation requires some degree of antigenic stimulation to detect the cells. And what is that we're going to use to stimulate the cells? One or a few epitopes, many, and there are certain issues related to this to his choices. So one thing is that is the antigenic complexity. And so to reiterate here, what T cells do recognize is a fragment, a peptide bound to HLA molecules. And there are many different, which is called epitope. And there are many different epitopes and many different HLAs. Here, for example, this is a study that we performed in allergies. And you see that there are many, many different allergens that are recognized in allergic asthma. And this is, I believe, this is cockroach allergies. And point is also that the pattern of recognition is different whether you look at people that have asthma or people that do not have asthma but are still allergic in terms of having rhinitis. So what happens here is that the antigens are different. And so, for example, if you were to measure an epitope derived from a green antigen, you would see a strong response in asthma, but no response in rhinitis. Or conversely, if you were to measure one of his blue antigens, you would hardly see a response in asthma. And so you would reach completely different conclusions because your bias in what part of T cells are you measuring. As I was mentioning, HLA molecules are what binds the fragment, the peptides, and presents them to recognition by the T cells. And that's how the T cells know that a cell is infecting in the first place. And there are many different HLA molecules. For example, in the case of class 1, there are 3 different molecules, A, B, and C, and 4 different molecules in the case of class 2, DR, beta 1, 3, 4, 5, DP, and DQ. And furthermore, beyond this polygyny, there is also polymorphism. So each of these molecules comes in many, many different alleles, actually thousands of different alleles. These HLA molecules, by the way, are the same molecules that are involved in HLA in histocompatibility. So these are the molecules that you need to match, for example, if you want to exchange a kidney. And as you know, it's difficult to find a match just because these molecules are so heterogeneous. So in the case of, for example, tetramers or assay methodology that use only one or few epitopes, sometimes people say, well, this gives me, this combination of epitopes give me 80%, 90% coverage. So what does coverage mean? So if you define coverage as the percent of individuals expressing at least one of the top 10 HLA molecules, for example, here in A, B, and C, then yeah, you would have 95.3%. But the trick is, or the complication here is that in having cover at least one of the top molecules doesn't mean that that is the molecules that that particular study subject participant uses. So if you're measuring responses, say, restricted by B702, and the donor also express B1801, A301, and 2701, does the patient know that he's supposed to use only that HLA molecule? You, again, may be missing a lot of the response. The other thing that makes things even worse here is that HLA molecules are polymorphic, and their polymorphism varies in different ethnicities. And there is a tremendous bias in the information that is available in the literature, because people study the alleles that are most frequent, and they're most frequent in Caucasians. So for example, you see here the number of epitopes that are known for A201 and DR beta 10701 is very, very large. But other alleles that are not as frequently studied, there is very few epitopes that are defined. And this is not because this particular HLA is better and has more epitopes. It's just that these are not studied. And so, again, a tremendous bias in coverage. So to overcome this kind of challenges, what we have been developing is what we call megapool approach. And so essentially, we try to be as unbiased as possible and combine in a large pool of peptide, which is generated by sequential lyophilization. So there's a few technical tricks to it. We try to cover as much of the potential or actual repertoire of fragments that are recognized to get an unbiased measure of the T cell specificities. And so one thing that one can do is just simply make overlapping peptides spanning the entire sequence of different antigens. And see, we've done that in the case of dengue, for example. So these are overlapping peptide pools spanning different antigens of dengue. Or in the case of SARS-CoV-2, we made overlapping peptides spanning the entire proteome. And we were able to measure responses like that. So this is applicable for instances in which you have a relatively good amount of cells, and also the target that you want to measure is not that great. In the case of situations where fewer cells are available, or the viruses are bigger, or whatever, there is another alternative, which is to make predictions for a large number of different HLAs and then combine the predicted epitopes in megapools of predicted peptides. Yet another approach is to, after something has been studied rather extensively, there is a lot of information that accumulates regarding the targets of adaptive immunity. And so you can just use experimentally defined epitopes to make megapools. And this is also obviously another approach that works. So depending on the particular situation, the target, the size of the proteins that want to be measured in terms of T cell response associated with that target, there may be different approaches. And we've done a lot of different megapools from different viruses, bacteria, allergens, autoimmunity, and so forth. And validated, of course, with patient samples. So now let me switch back and go into the second part of the talk. And this is the slide I showed you earlier, and depicts the three different types of responses associated with adaptive immunity. The antibodies, the neutralizing antibodies that are key to prevent infection, the helper and killer T cells that are key to modulate disease severity and terminate infection. So we were very interested in determining the type of immune responses associated with SARS-CoV-2 infections. And because of our bioinformatic expertise, we also run a database for the NIH called VIDP, where we catalog all T cell reactivity and we can predict epitopes and so forth. We were able to jump on this very early on, and we were able to generate this first set of data. This was the first paper that described early on adaptive responses to SARS-CoV-2. And the issue was very heavily debated because at one end of the spectrum, some people were fearful that an overactive T cell response was responsible for the acute respiratory distress syndrome, so it was killing people. At the other end of the spectrum, some people were concerned that the coronaviruses in general are not very immunogenic, and so this was not going to be good for vaccine development and so forth. And so this was the first study, and what we did study was T cell antibody responses in mild cases of COVID-19 after the people were recovered. And this was done by design to really understand what does a success scenario look like? What does an immune response, a successful immune response look like in someone that was infected and did not require to go to the hospital, were able to deal with the situation on its own.
And also a paper from NIH grantees from La Jolla just came out in the journal Cell.
And this was Dr. Tony Fauci in the spring in a congressional hearing highlighting our paper. And what did we find? So we found that there were good antibody responses against spike, both IgG and IgA. And we also found that there were good CD4 and CD8 responses also reacted against spike. And this was very important because at that point in time already people were developing SARS-CoV-2 vaccine based on the spike antigen. So it was good news, it was reassuring. And at the same time, we also detected responses against other antigens, which raised the possibility, but if a broader spectrum of T-cell response was desired, these other antigens could also be considered. Also, these other antigens are much more conserved than spike, and so are of particular interest in the context of variants or also potentially a broad coronavirus vaccine. So what we could not do in the spring of 2020 was to answer the question, how long does the immune response last? Because as an immunologist or vaccinologist, to get to the memory phase, you have to wait about 6 to 8 months, and then you want to see how the immune response pans out. And so this was followed by this other study that came out in Science, eventually in Press in early 2021, but was available beforehand. Essentially, we waited as soon as we reached 6 to 8 months time point of the patients that we were following. We ran additional studies, and we found that in reality, a good fraction of the vast majority of the previously infected people still had a sizable response at 6 to 8 months, at least in some component. So that was good news. At the same time, glass half empty as full already indicated that the immune response can wane over time. And also, based on that, we were very vocal in suggesting that people that had been naturally infected should nevertheless get vaccinated as soon as vaccines were available. In fact, it turned out that people that were infected and then vaccinated had this so-called hybrid immunity that generated an immune response which was superior to what you would see in natural infection alone or vaccination alone. Now, what is the evidence that T cells are actually doing something in SARS-CoV-2? And there are several lines of evidence that are summarized in some of these studies. So after we did the convalescent mild disease studies, we immediately turned our attention to the acute phase. And we asked a very simple question. If I look at early responses in acute phase, what is the best predictor of a mild disease versus a severe disease? And what we found is that the antibodies really did not predict that. What predicted a mild, less severe disease was having early CD4 mostly and CD8 T cell responses. Again, this is consistent with the notion that the T cells, having an early T cell response is really what is going to control the disease and impact disease severity. This data was confirmed by Bertoletti's group in Singapore, but added an additional very important piece of information that the early T cell responses were also resulting in less viral load. We recently also produced another study from early samples from 3 different Italian sites, wonderful collaboration there. And what we saw, again, we confirmed the data that the early T cell responses were associated with less severe disease. But in addition, we're able to show that people that had a broader response, so not just to spike, but also to other antigens, again, had a were associated with a less severe disease pattern. There is evidence in animal model of disease. I'll talk about it in a second. And also, we're going to mention that in another set of slides. There is evidence in humans that have absent or decreased antibody responses. These are either people that have X-linked agamma-globulinemia, so these are genetically impaired in their capacity of producing antibody responses, or people that have, because they are MS patients or patients for hematologic malignancies, that they have been treated to essentially wipe out their B cells. And in many cases, these people can actually have a mild disease course following vaccination, again suggesting that in absence of antibody responses, T cells can control the disease. This is obviously not to say that antibodies are not good. Antibodies are good. You want antibodies. It's just to say that even in absence of antibodies, you can have a mild disease outcome, and your body can control the disease. So I'm not going to go through this in any detail, but just it's a lot of data in animal studies. Even before COVID, studies from Perlman group in MERS and SARS-1 showed roles for CD4 and CD8 T cells. Lots of study in murine models, mouse models of SARS-CoV-2, and very importantly, in studies in monkeys, it's non-human primates, mostly by Baruch's lab. It really showed that depleting CD8 T cells in particular was associated with trouble from the animals to control the infections, particularly in condition of limiting antibody responses. Again, highlighting and demonstrating a key role of T cells in SARS-CoV-2 infection. So this brings us back to this concept that if we want to protect against infection, really you want antibodies. Those are neutralizing antibodies are a key component, and maybe a minor component also from cellular immunity and T cells. But in protection against severe disease, in protection against hospitalization and death, really the role of cellular immunity becomes prominent. So as the vaccine became available, we did studies where we characterized immune responses to different vaccine platforms. Here I'm just showing you the mRNA platform to make a point. And the point is that when you look at immune memory to COVID vaccines, you see the antibody response goes up very quick and then wanes by about a log already in a 6 month period. But if you look at for example, here the CD8 response, the waning of cellular immunity is much slower and much more limited over, for example, this first 6 months following vaccination. This actually fits with the data that was coming out, and this is before variants. And you see here and this is not our data, this is a Kaiser study, but you see that the protection against infection waned over 5 to 6 months. But even in the same study, you look not at protection against infection, but protection against hospitalization and death, the protection was still maintained. So what about variants? Going back to what we were discussing here, the concept that what T cells recognize are these fragments, these epitopes bound to HLA molecules. And there are hundreds, thousands of different epitopes recognized in human by CD4 and CD8. And because HLA molecules are different from an individual to the next, typically the epitopes recognized by one person are different from the epitopes recognized by another person. And this number now it's about over 3,000. So there really are thousands of different epitopes. So what this means is that it's very difficult in general for viruses and for SARS-CoV-2 in particular to escape T cell recognition, because they would have to mutate tens and hundreds of different epitopes. And even if someone may have a virus may accumulate mutations to escape the immune response in one person, those mutations would not do any good infecting the next person, because again the HLA molecules are different. So this is very different from what you observe in the case of neutralizing antibodies responses and neutralizing antibodies interfere and block the particular region of spike, either directly or by steric hindrance, particular region of spike that spike uses to infect the cell. And so by definition, the sites that the virus can mutate and does mutate to escape antibody recognition are more limited. So it's relatively easier for a virus to escape to mutate antibody recognition. So we published an early study where we showed that really the T-cell response was largely preserved in either infected or vaccinated individuals in the early variants that were detected. And this was followed up by another, and this was Dr. Fauci showing our data at the meeting, a briefing at the White House. This was followed by other studies where we did a much more comprehensive analysis. Now, of course, there were Delta, Lambda, Omicron, but we saw and we studied responses to people vaccinated with mRNA, with the J&J, Janssen viral vector, Novavax, and we looked at early, intermediate, memory phase, and the result was always the same, that basically while the antibody response was significantly impacted by the mutations found in the variants, the T-cell response was largely preserved. In 80–90% of the response was still preserved at the 6-month post-vaccination point. Bioinformatic analysis provided a rationale for why this was happening, is that in reality, 90% of the epitopes were totally conserved in the various variants. And this jived with the observation that when you looked at different individuals, each individual had a very broad response with many different epitopes being recognized. And so that even if one was impacted by a mutation in the variant, that was not leading to a substantial decrease at the population level. We can see occasional people that have a significant decrease in T-cell activity, but at the population level, really the vast majority of the response is preserved. And this was confirmed by collaborations with different groups, fantastic collaborations with people in South Africa, the Netherlands, Sweden, Switzerland, showing the same thing. And so really verifying that the same thing is true in both patient and vaccinee cohorts from different ethnic groups, different geographical locations. This is just a quick slide to highlight the fact that this in reality is nothing new. The fact that it can be cross-reactive recognition of variants is well documented in the influenza system, where it's also shown that this cross-reactive T-cell recognition may play a role in modulating disease severity. So expanding the concept a little further, it's interesting to consider another dimension, which is not so much the cross-recognition of SARS-CoV-2 variants, which again is a situation which fits with what we're seeing in the sense that the antibody response is decreased, the T-cell response is preserved, and a lot of the variant waves have been associated with very large number of infections, but the vaccines have largely maintained their protection against severe disease and death, and we'll get back to that. So, but this is a further extension of the concept in the sense that maybe even some cross-reactivity beyond variants also with other coronaviruses, for example, common cold coronaviruses. And this goes back to the early studies in the spring of 2020, where we saw that many unexposed subjects, which were actually blood donations obtained before the start of the pandemic, they had a pre-existing immune memory, T-cell immune memory. We went on in a different study to show that this was, in at least some cases, really clearly mapped to common cold coronaviruses that had very significant homology. So basically, what happens is that if there is a T cell very specific for a certain sequence in a common cold coronavirus, and that sequence happens to be largely preserved and conserved in SARS-CoV-2, those T cells can actually recognize SARS-CoV-2, even though that particular person has never seen SARS-CoV-2. In fact, in this study, we showed that the common cold coronavirus sequence was recognized even better than SARS-CoV-2, suggesting that the original stimulus that elicited that memory T cells was common cold coronaviruses. We further showed in another study that in reality, the people that had this pre-existing memory developed better, faster CD4 T cell responses and better and more durable neutralizing antibody responses. There is a lot of data that has accumulated over time to show that indeed this pre-existing cross-reactive response for T cells is beneficial both in natural infection and in vaccination. This excellent study from Thiel's group. We also studied reactivity in healthcare workers, showing that healthcare workers that, because of their occupation, are heavily exposed to common cold and other viruses, had an also heightened cross-reactivity, pre-existing memory T cell, and a number of excellent studies from Lalvani and Bertoletti and Maini showed that indeed this was consequential at the level of disease outcome and potentially having a role in preventing infection, having a link to a symptomatic or abortive infection. And this other study in JCI showed that indeed a recent endemic coronavirus infection was associated with less severe COVID-19. Very impressive data. So finally, in terms of assays to detect T cell responses, I showed you a lot of data that was generated with AIM and ICS assays. Lately, we've been studying a lot of breakthrough infection, trying to understand what is breakthrough infection and how it's linked to disease outcomes and what originates the different outcomes and so forth. And so we wanted to develop assays that would discriminate people that have cross-reactive responses, but in reality, they might have pre-existing immune memory, but were never infected nor vaccinated, with people who were infected but not vaccinated, or people that were vaccinated and not infected, or people both. And so our simple notion was, what if we measure spike and the rest of the genome in terms of targets of T cell responses? We should be able to see differences. And in fact, we do. So here you see, these are the T cell response against spike, and this is a T cell response against the rest of the genome. So people that have never been infected nor vaccinated have pre-existing memory response, and they are this gray ball. You can set appropriate threshold to weed them out. People that have been vaccinated but not infected, we tend to see, of course, only spike and not the rest of the genome. People that have been infected and but not vaccinated, because they have seen the virus, they obviously see both spike and the rest of the genome along with diagonal. And people that have been infected and vaccinated, they also see both spike and the rest of the genome, but they've seen spike twice in vaccination and infection. They've seen the rest of the genome only once. And so they have a higher component of spike in their response. And this can classify these 4 different disease status with an accuracy of over 85%. And this is what I was mentioning earlier, the fact that in many instances, people that have decreased or absent antibody response can nevertheless develop a good T-cell response upon vaccination or after recovering from infection. And this is very important. And it would be important to have standardized assay to detect these T-cell responses and link them to what level of T-cell responses may be associated with protection from severe disease. At the end, this is the picture we're getting. In an unvaccinated individual, we have a lot of different potential disease outcomes, from asymptomatic to hospitalization and death. In someone that was recently vaccinated and a pre-variant situation, we all remember the vaccines giving 95% protection against infection, but protection waned over time. Variants came to escape the neutralizing antibody response, and so the protection against infection decreased, but still the vaccine have been holding their grounds against hospitalization and severe disease. There are ways to reset the clock, so to speak, by boosting or hybrid immunity, and also updated vaccines, perhaps including variants or including other components of the viruses, or including nasal delivery. This concept is here, you see the of dissociation between positivity and death. So this graph shows the overlay of positivity and deaths in data from the CDC until the December 2022. And you see that in early stages, the two tracked fairly well with each other, with the deaths being offset by a few weeks because of the time that is associated with the disease development. But then, after the Omicron 1 wave, we really started to see these dissociations, that people were getting infected that were not getting sick or dying as much. And in fact, it's difficult to estimate now infections because tests are no longer reported. Everybody gets a home test, but if someone is positive by a home test, it doesn't call the CDC to report that. A more accurate measure here is given by wastewater. And this is, again, a San Francisco wastewater analysis showing the dissociation between COVID cases and positivity in wastewater, again suggesting the dissociation between infection and disease severity. We are building an immunity wall, and there are two components that come into the immunity wall. One is repeated booster vaccinations. This is from Eric Topol's blog, showing a number of different studies clearly showing the benefit of multiple vaccination. This is another study instead showing the benefit of having had a previous infection protecting against a subsequent infection with a COVID Omicron variant. The immunity wall, though, is not necessarily as strictly dependent from the number of infections, but also from the time from when the last booster was received. But nevertheless, this is a data from an New England journal, very interesting, shows you the protection from severe or fatal Omicron infection. If you had previous infection or 2 doses of vaccines, you're here in the 70%. 2 doses and 1 infection or 3 doses puts you higher, and 3 doses plus a previous infection gives you even higher protection. So at the end of the day, get a booster, especially if you have not had an infection or a vaccination in the last few months, certainly 6 months, but I would say even 3 or 4 months. What is going to happen next? I don't know. Depends on many variables. One, are there going to be new variants, and specifically as new variants evolve, what do they look like? Are more or less virulent and dangerous? Of course, we need to continue to monitor the situation, because otherwise we would not know that variants are evolving and which variants, and also to develop countermeasures, updating and generating second generation vaccines. And it's possible that we eventually may evolve towards a steady state of relatively stable immunity. In fact, we have published this recent study looking over a 3-year period, not at COVID, but at the closest human relatives, the common cold coronavirus. Of course, there is no guarantee. SARS-CoV-2 is a different virus than common cold coronavirus, so there is no guarantee that that's the situation we'll end up in, but it's a possibility. And so, sorry for this slide, it's a little blurry, but I just wanted to re-emphasize the concept that it's really two different things if you're talking about protection against infection, where there's a major role of neutralizing antibody, or protection against hospitalization, severe disease, where really the T cell and cellular immunity play a key role. So the protection of infection really requires a high dose, high levels of neutralizing antibodies, but there's this zone where different components contribute to protecting, not from infection per se, but from disease. And with that, I just want to close saying that everything I showed you is the result of a close collaboration between my lab and the lab of Shane Crotty, strong contributions from a number of people in this lab, and from our Grifonian instructor at LJI, Ricardo Silva, also instructor at LJI, and Professor Daniela Weisskopf, an assistant professor at LJI, and many, many other people. And with that, I would like to thank you very much, and we will move into the question section.
Thank you very much, Dr. Sette, for this fantastic talk. And now we have time for around 15 minutes for questions. We receive questions from the audience, and there are questions about generally for T cell response, and then questions for SARS-CoV-2. So I'm going to start with the general questions. Okay?
Very good.
Perfect. So the first question is, what is the advantage of measuring T cell response over antibodies?
Well, it's not an advantage per se, and certainly I think that both antibody and T cells should be measured. So the answer is that really gives you a more complete picture. Our studies and studies from many others have shown that you can't really predict how many T cells or how many memory B cells you have from measuring the antibody responses. And perhaps the most striking example of that is that as the neutralizing antibody titer goes down, the number of memory B cells goes up continuously over the course of 6 months. So in the context of protection against infection, yes, measuring antibodies is relevant, but really doesn't tell you necessarily how protected are you going to be against severe disease. One particular other situation which is very striking is the situation of people that have B cell deficiencies, and so they don't make antibodies. They might have B cell deficiencies because they simply are genetically that way, or because they have been treated with anti-CD20 because they have MS, or because they have an hematological malignancy. In those cases, people still get vaccinated, should get vaccinated, and in fact, they do mount a T cell response that has protective effects. So that is as an example of how both provide meaningful information.
Great, thank you. The next question is following up on your letter to the FDA. Could you please share more about their feedback?
I was not the main signatory. I was one of the signatories. And today I am not aware of any specific feedback from the FDA. Now, I'm not saying necessarily they should and provided a feedback, because that letter was more of a plea, a suggestion for considering that. So it's quite possible that the letter was received in part or in all, and maybe next time there is a process of regulatory approval or something like that, they might consider some of the points that were made in that letter.
Great, thank you. Okay, can we use mass spectrometry-based assays to measure T cells?
No, mass spectrometry assays do not measure T cells. They're very useful assays, and what mass spectrometry does is essentially detect whether one particular peptide fragment is generated in that particular cell. So in a way, it does give you information about whether the peptide binds to HLA, because otherwise you wouldn't find it bound to the HLA, and whether the peptide is generated by cellular processing. So very interesting information. However, there are also limitations. One limitation is that it's done in some kind of cell line, usually, or in PBNC pulsed with something. And so it's often unclear how physiological that is, because the peptides or the situation in the lung or in the lymph nodes may not reflect the cell line that is used. Is also the cell line is one HLA haplotype and maybe not representative of the full spectrum. And also unless this is done with infectious virus, also infection in vitro may not mimic infection in vivo. So there's lots of different things. But the most important thing is the fact that a specific peptide is generated by processing and binds to HLA. It does not mean that that peptide is going to be recognized by T cells. In fact, there's plenty of peptides that are generated by processing and are not recognized by T cells. In fact, all the self-peptides, it's an issue not as much for infectious diseases, but for cancer immunology. There's lots of peptides in a cancer cells that are not necessarily recognized by T cells because of tolerance, because of suppressive environments, and so forth. So very good assay, very informative, not necessarily informative per se of T cell recognition.
Great, thank you. So the next question is, do you know about the T cell response in Lyme disease? Is there a clinical utility to measure T cell response in Lyme disease?
I have not studied Lyme disease, and so I would be hesitant to go out on a limb on something I really don't know much about.
No problem. Okay. So how do you envision the future of T cell testing in infectious diseases? Will more assays be FDA approved?
I'm not the FDA, and I cannot tell you what assays might be approved or not approved. I think that there is certainly a need for T cell assays. There are a number of groups we have published, and others have published on a interferon, kind of quantiferon whole blood cell assay. Other people have also been exploring ELISpot assays as a assay to measure T cells. There are other assays that are in development. The Bertoletti's group and Mount Sinai, I believe, have an assay where they measure mRNA changes and so forth. So there are a number of assays that are of interest and are being developed, and so it's an area that is moving. There still remains a fundamental problem that T cells are more difficult to measure, and so easier assays usually give less granularity of information.
Great. So now we are going to move to the SARS-CoV-2 specific questions. And the first one came during your first slide, and it says, in your first study, you observed good IgA and IgG responses. What about IgM?
To be honest, I don't remember. I think we measured them, but in a memory type of situation, IgM tends to be less informative because they come and go. And so what you really want to look at is IgG and IgA. But I confess we might have measured, and if we did, it's published, but I don't remember.
Yeah, I know from other studies that IgM response is not as strong as IgA. So maybe that's why that could be one reason.
Well, and also IgM in general is transient because it's the first type of antibody response, but then luckily for us, the B cells switch to IgG or IgA. So it's, yeah.
Great. Okay. Second question, you mentioned that early T cell response controls the severity of the disease. Have you seen this with other infections, or is this specific for SARS-CoV-2 infection?
So certainly there are many examples of this phenomena, and I can mention two very interesting examples. One is actually topical today, these days. It's pox virus infections. So in the case of pox virus infection, again, clearly antibodies are what is crucial for protection from infection and prevention of disease. But it was very much apparent when the old smallpox, the Dryvax vaccinia virus, was used in the general population. People that are deficient in T cells actually they get sick because they have a very hard time in controlling the infection caused by the live attenuated virus. And so that's why the Dryvax vaccine, which was perfectly good vaccine and very effective, was discontinued because of the issues with people that were immunocompromised. So there is very clear evidence in the case of a poxvirus infection of the T cells really being crucial for the control and the termination of disease. Another example, which is worth emphasizing, is the case of flu. You recall the swine flu of 2008, the pandemic in 2008. So what happened there is that it was noted that there was this strange severity of disease curve. Normally, older people do worse in general because that's unfortunately us old people are usually less effective immune responses. In the case of the pandemic, the trend was opposite. Older people was noted tended to have milder disease. What was also noted that coincided with the fact that decades earlier, another H1N1 had circulated in the human population. So people that were old enough to have a memory response that still was there, again, did not escape getting infected, but were not having as severe of a disease.
Okay, great. Thank you. Now we just received a question from the audience. Have you noticed a different T cell response depending on the type or diversity of COVID-19 vaccine, homologous versus heterologous vaccination?
So we have not done that very much. We have done some studies, but certainly there is a lot of data showing that homologous and heterologous vaccination matters. So if you're talking about comparing different vaccine platforms, yes, we published that study that I mentioned where we compare 4 different vaccine platforms. There are other studies that came out that also examine the response of inactivated viruses and also looked at AstraZeneca and so forth. So there is a lot of data in terms of comparing different vaccine platforms. And as I was mentioning, the mRNA vaccines have much higher antibody titers as far as we can tell. They do wane over time, but probably they tend to kind of plateau out. And the viral vectors do not induce as much neutralizing antibody response. They tend to be a little bit more stable. All the different platforms have been good in inducing T cell responses at the CD4 level. Adjuvanted protein or inactivated vaccines are not as good as inducing CD8 killer T cells, which is also to be expected. So lots of data there. In terms of the heterologous prime boost vaccination, that's a fascinating subject. And the data clearly shows that in some cases, the heterologous booster vaccination does give you very good results. And that I've seen is mostly in viral vector followed by mRNA. And interestingly, if you do it the other way around, it doesn't work very well. And that is not very well understood. Viral vector with viral vector boosting doesn't work very well at all. And that might be perhaps due to antiviral vector immune responses that build up over time. But that is just a speculation. I don't have any data to really comment on that one way or another.
Okay, great, thank you. Have you seen any patients with no detectable antibodies and no detectable T cell response? If yes, did the patient have a severe disease?
Well, so I guess the question is, obviously that would be a patient that is incapable of mounting upon infection a B or T cell response. I don't know that I've seen that. That would have to be such a profound immunocompromise that I don't think it would be very common.
So it's not common. Okay, thank you.
I mean, I don't think that happens.
Yeah, no. Great. For a patient with lymphoma, how could their body respond to the COVID vaccine?
Well, that goes back to what we were saying earlier. People that are generally immunocompromised for both B and T cells, they tend to respond less well to vaccination or infection. And that's why it's actually common practice to give more vaccinations, more boosters to people that are immunocompromised. And in that respect, that is also bringing out an important consideration in terms of boosters. So clearly, certain segments of a population, like immunocompromised or old people and so forth, should really get as boosters and continue to get boosted because that's very important to protect them. Depending on what kind of lymphoma and what kind of immunocompromise, as I was mentioning, people that have a defect in B cells can nevertheless mount a T cell response, and that is of value.
Great. So following up this question about immunocompromised, would you recommend testing for T cell response only in immunocompromised patients or in all patients?
Well, that is a question that opens another question in the sense that, yes, it would be great to test everybody for T cells. The problem is that it's really not clear what level of T cell response or what level of antibody response is linked to protection. And I get this question all the time. I get questions like, oh, I had 3 boosters and I was infected 8 months ago, and my antibody levels are this. Should I get a booster? And I say, well, I don't know. I mean, I don't know how to interpret your antibody levels. And if you have been, it's been 8 months since you just got an infection or a vaccination, probably I would get a booster, but also depends in how old are you? Do you have other underlying conditions? And are you going to be going to lots of crowded places? Are you in a place where there is a current wave of infections going on? So it becomes a little bit more nuanced. In the case of immunocompromised patients, well, that is there are studies ongoing where people are trying to correlate patient groups that have different level of T cell responses with are they protected or not. I don't think that the results are out, but if there was a threshold that would say, aha, if you have this much T cells, then you are likely to be protected against not from infection or from severe disease, that would be of value, because that would say if you're below that threshold, maybe you should get another booster, or in any case, you should be super careful with your masking and with your social interaction and so forth. Well, if you would be above that level, you might have a higher degree of confidence. But right now, that level has not been defined.
Okay, great. Thank you. And we have time for one more question. Then the other ones we will address via email. And this question is a follow-up of the previous question. So could you please share your thoughts on why testing for T cell response in COVID-19 patients is not recommended in guidelines? Will it change in the near future?
Well, that goes. I don't think it's going to change in the near future from what I see, but I would be happy to see some more action in T cell testing. And I think the difficulty is related to the issue that I was mentioning earlier, namely that there is no clear T cell threshold that is associated with protection, and also that is bound to be influenced by the level of neutralizing antibodies and the general patient populations that is considered.
Okay.
So I see this as a additional element of information. In a way, like measuring T cells in tuberculosis, it's done routinely, but it's not taken as a per se as a diagnostic. It's taken in context, also other assays to interpret the situation.
We have one more question from the audience that I think is important to ask you. When we will see T cells that can be used on patients in hospital clinical microbiology labs?
I do not have an answer for that. I think we'll have to wait and see.
Perfect. So thank you once again, Dr. Sette, for your time today. It was a pleasure to have you here. It was a great talk. So thank you. And thanks everyone for joining us today. Today was the last webinar of 2022, but we are working on more webinars for 2023. So please stay tuned and have a wonderful rest of the day. Thank you very much and see you next year. Thank you.
Thank you. Bye-bye.
Bye.